So today we will open up a new topic, DNA repair mechanisms and cancer. This is a very important topic, as you know. I have always told you that most of the anti-cancer agents, they act through damaging the DNA. So it becomes a balance between our DNA repair mechanisms and the anti-cancer agent. Whenever there is some DNA damage, the DNA repair mechanisms will counter that. They will restore the DNA to its original shape. So one has to overcome that in order to induce killing in the cancer cells. So in that context, it is very important in cancer. So when does this DNA repair mechanisms operate? Obviously, whenever there is a damage to DNA. So the next question is, what do you mean by damage to DNA? What kinds of damage occur to DNA? So those are the important questions. The permanent alterations in the DNA structure, you call them as mutations, and the accumulation of these mutations naturally help in carcinogenesis. They can ultimately, the normal cell can become cancer cell if it accumulates several mutations. That is what we know. But then sometimes the DNA replication and other processes, the cell cycle that you have studied, these mechanisms are not 100% error proof. There can be some mistakes in your cell cycle proliferation, the regular mechanisms. They are not error free. So spontaneously also certain changes occur in DNA, spontaneous changes. We have studied a little bit about that, even there was a question in the quiz. Cytosine residues can undergo spontaneous oxidative deamination to form uracil. This may occur at a slower rate. Similar, another change, I am listing some changes. One is the cytosine changing to uracil, that is one change that can occur spontaneously. Hydrolysis of N-beta glycosylate bond, I hope you know what is a glycosylate bond, between a purine pyrimidine base and the sugar. The bond that is there, you know, in the nucleotide, the base, sugar and phosphate are there. So that bond between purine pyrimidine bases and the sugar, that might get hydrolyzed. That means they get separated, they are linked and they may be delinked. And this is happening at a slightly higher rate than what I mentioned earlier for purines than pyrimidines. That means purine being linked with sugar may get hydrolyzed at a faster rate compared to pyrimidine getting linked to a sugar. In the case of RNA, this is still slower. So these are all some observations people have made. Now we know very well that radiation, environmental agents such as radiation, chemicals, they can cause DNA damage. All of us are aware of that. But we are only just systematically studying. When there is UV radiation, if it causes DNA damage, two types of thymine dimers are formed. Thymine is the residue that is usually affected. It's called a thymine dimer. There are two types of thymine dimers which are shown here. You can look at it. That is this particular case. If you look at that, this is the thymine part, and this is also the thymine part. This is the sugar and phosphate. It's linked, right? It's very easy to make out. But you see the particular linkage that is happening. You see the linkage is between the carbon, 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 carbon here. So it creates something like a square with 4 carbon. What do you call this? Cyclobutane. Cyclobutane. So that this dimer, thymine dimer, because there is one thymine here and one here. So they form a dimer. So naturally this will prevent further division, isn't it? The DNA will get damaged and the base pairing and all that will be definitely affected, right? This is one damage. There is another dimer that is formed. This is something like a 6 carbon and the 4th carbon will be linked. It's called a 6,4 photo product. Photo because the light is involved, radiation is involved. So that is what. So these two types of dimers, you may ask what they will do. They will introduce a bend or what we call sometimes as a kink in the DNA. So the DNA structure gets altered. So that is what you call as a DNA damage, right. So when there is UV radiation, these are the possible changes. Gamma rays and X-rays also can damage. They can fragment. So you see there can be some mild damages, there can be very serious damages also. Another interesting feature that you may have to think about is when DNA is a double strand, so there can be a single strand break or there can be a double strand break. Which one will be easier to repair? Single strand, obviously, because if the two strands are broken, then it becomes. The one reason for that is when the single strand is broken, you have the complementary strand alive. So that can, with that, if you have the necessary enzymes, you can resynthesize the other strand after damage. So that is why single strand breaks are somewhat easier to tackle by the system DNA repair mechanism, but whereas double strand breaks are a little bit difficult. But still you have mechanisms. There are as many as 6 different methods by which you can damage. There are about 3 methods for the single strand, 3 methods for double strand. There are actually more methods, but the more prominent methods are around 6 methods. We will look at them. So the second one, gamma rays, X-rays, they can break open the, here if you observe the UV rays, the ring was not broken, right. It was only attaching with some other, the 2 thymine rings are actually attached, whereas it can be completely broken also with gamma rays and X-rays, thereby fragmenting the DNA bases after opening their rings and breaking the covalent links. So that is what happens sometimes. Similarly, chemicals can do this some kind of damage. One chemical is nitrous acid. Nitric acid, you know, HNO3. HNO2 is nitrous acid, and some alkylating agents. This nitrous acid is not totally something very poisonous. It is used as a food preservative. It all depends on what is the dose you are using. I mean, the concentration or whatever, the amount. Ultimately, in trace amounts, if you add in the food, it is acting as a preservative. That is why it does not pose a big threat, because of small quantities that are used in the food preparations. Alkylating agent such as the dimethyl sulfate, it methylates the guanine residue to form what is called O6 methyl guanine. You can see here, this is the guanine structure. I hope you know. Guanine is what? It is a purine. Adenine and guanine are purines. Cytosine and thymine or uracil are pyrimidines, you know that. So that is not an issue. So you have this naming, I mean numbering 1, 2, 3, 4, 5, 6. You see it's written usually these purines are generally, the nomenclature is like you give the numbering starting from here. You see 1, 2, 3, 4, 5, 6, something like that. They have given, it starts from here probably 1, 2, 3, 4, 5. How it is coming? Just see. Clockwise or anti-clockwise? 6, 5, 4, 3, 2, 1. Yeah, it is clockwise, because it's just, is it clockwise or anti-clockwise? Yeah, clockwise only. So it is the 6th position. This one is 6th position. So once you remove that and attach, what is this group? O methyl group. O means oxygen is there. So here you are breaking this bond and attaching a methyl group, O methyl. So 6 O methyl guanine. So this is one change with the, when you add dimethyl sulfate, this is what happens to the guanine residue. So guanine residue gets modified, just like the thymine residue got modified with UV. Here guanine residue gets modified. I am taking some few minutes to explain this because this has got importance in disease conditions. That's why. Uh, Another thing is hydroxyl radicals. Now, what will happen that 6-methyl guanine, ultimately the base pairing will be affected. Pairing with A base pairing with the T, G base pairing with the C, you know. So, that G with C, that base pairing will be affected. Hydroxyl radicals can also cause DNA damage. Reactive oxygen species formed by our cells, you have many enzymes to tackle. What are the enzymes? Superoxide dismutase, catalase, peroxidase, so many enzymes are there. But under some conditions, they can also cause damage. If there is a reactive oxygen species, excess reactive oxygen species, they can damage the membranes, the lipid components of the membranes, and ultimately they can also damage the cells. As I already mentioned, the normal DNA replication process has some errors, can generate errors, such as we will discuss this mismatched base pair and all that. This can result in mutations such as insertion, deletion, substitution, all this. I hope you know what are these mutations. Frame shift mutation, deletion mutation, all those nonsense mutation, missense mutation, these things you should have studied. In one day, there may be thousands of such changes occurring in our cells, but DNA repair mechanisms see to that. What is the need for DNA repair mechanisms? As I just mentioned repeatedly, DNA replication itself is not a perfect process. There may be errors, but that is set right by our DNA repair mechanism, so that you have only very minimum errors, like it is given some statistics here. Fewer than 1 in 1000 becomes a mutation. You know how many DNA pairs are there. So, out of that 1000 is a small number comparatively, because you have so many nucleotides. So, 1 in 1000 becomes a mutation, and thus making DNA as a relatively stable molecule. If DNA repair mechanisms are defective, the damaging reactions, what will happen? They will accumulate, and this will aggravate the situation. Ultimately, it can give rise to some kind of genomic instability. So, that is what ultimately this helps in cancer progression, tumor progression. That is what is shown here. DNA, the main thing in the DNA repair, what is DNA repair? DNA repair means DNA damage alters the structure of the DNA. DNA repair restores it to the normal position. That is all. So, DNA damage means it damages the structure, it alters the structure. DNA repair means it restores the structure. Simple. This is what it is. And how to restore? By simple Watson and Crick base pair rules. So, G will base pair with C and A will base pair with the T. You must have those nucleotides and you must have the appropriate enzymes, ligase, DNA polymerase, all those kind of enzymes, and ultimately you can put them back. So, that is the simple idea. So, single strand breaks, we have an advantage in the sense that the one strand is damaged, the other one can act as a template, which I have already told you. Many anti-cancer agents induce DNA damage, that I have already told you. What are those anti-cancer agents which are prominently doing this? One class of anti-cancer agents are alkylating agents. Some examples I have given: cyclophosphamide, 1,3-bischloroethyl nitrosourea, BCNU as it is abbreviated, and then you have this platinum based anti-cancer compounds, cisplatin, carboplatin, oxyplatin, oxal is written oxaliplatin, so like that, and adriamycin. So these are all the famous alkylating agents which you should know because this is very important. Then double strand breaks can be induced by, we have already seen, I told you, gamma rays and X-rays can do that. Severe damage can be done, but apart from that, ionizing radiations and bleomycin can do this. A combination of these drugs may cause more damage than when a single therapy is used, suggesting some kind of synergy. That is, if you have two agents that can induce DNA damage, if you combine them, naturally you will get more effect. So, after all, you for treatment ultimately you also require to overcome the DNA repair. In that sense, what will happen if you have chemicals that will block DNA repair? Will it be helpful for the therapy or will it be against the therapy? Helpful. So, what they do? They sensitize the cells. So, that is also what is emerging is that a class of sensitizers that are merely DNA repair inhibitors. There will be slight confusion if you think in the wrong directions, because normally when you have in the normal cells, when you have the DNA repair these things inhibition, then there will be DNA damage that will lead to cancer, right? But when these things are happening in the excess, actually more DNA damage if you do, if already cancer cells are there, then it is advantageous to us. But if you are thinking from, if your starting point is normal cell, this will give totally opposite reaction. This is why we get all confusions in biology. It depends on the microenvironment and the stage. That is why the same everything is a double-edged sword in biology, more or less. Same thing will do killing, same thing will do survival also. Same compounds. That is because it depends on the ultimate environment. That's what I told you. Oncogene is a growth promoting gene, but if you overexpress oncogene, it can kill the cells. You remember that example. So that is why it depends. Here also the DNA repair also, sometimes even I may get confused if you ask something about it. Because if we, for a moment, if we forget it's a normal cell or cancer cell, then we are bound to have confusion. Keep that in mind. These compounds work better in combination with, that is what we are talking about is DNA repair inhibitors. They will work better in combination with traditional DNA damaging drugs. So, you can just keep that in mind. Now, we will pause, we will systematically study what are the DNA repair pathways. As I told you, there are a number of pathways, more than one. We will also talk about that. If you have more than one pathway, is it advantageous or disadvantageous? If you have more than one DNA repair pathway, now we are talking about normal cells. So, do not worry about the cancer cells. That is where the confusion will come if this question is asked. Advantage. What advantage? Correct. Spare bus. You want to go somewhere, you have two cars, one car something happens, so you have another car, you can take that, right? Or bus. If you are poor, you can take cycle also, it's okay. But whatever. So, spare bus, something like that. So, you have a spare to meet your needs, additional thing. That is one idea. Sometimes the cells may be defective in one pathway, and so if they have another pathway, they will now normally they may respond to a signal by one particular pathway, but now they will depend more on the whatever is available, that particular pathway. So, that is one way it is helpful. We will see the other things later. So, just there are different pathways. They are called base excision repair, BER, nucleotide excision repair, mismatch repair, homologous recombination, non-homologous end joining, and then transletion DNA synthesis. For understanding all this, you must have some good idea in molecular biology, deletion, insertion, all those things you just read up. But basically you understand that we all are born from a fusion of sperm and ovum, and we get one allele from the mother and one allele from the father, right? So we will have two copies of the same gene. This is an important concept for you to remember. Then only you will be able to understand the homologous and non-homologous end joining, because you have another copy there. We were talking about a single strand break, we said the other strand is there. Imagine if it is a double strand break, both the strands are broken, but you have already two alleles. If only one copy is broken, if both the copies are broken, then if you imagine like that, then it is difficult. But only one copy is broken, damaged, then the other copy is there. So that is when that even double stranded DNA damage, the system tries to help. I am giving an overall idea, I will go one by one. The other situation is, do you want to always completely restore the structure? Sometimes you are compromising in life. After all, cancer biology, cancer treatment is full of compromise. We know the drugs given are deadly poisons, still we are administering them to patients because we see the advantage. Some disadvantage like losing the hair, some losing some immunity, something like that, we are compromising. So we have learned to compromise in every part of life. Similarly here also, some of the repair mechanisms may not restore it completely. That means if you have, let us say, 20 nucleotides you have lost due to DNA damage, maybe put them back some 10, 50%, you are happy. So like that, some compromise is also there in some of the pathways. Mostly they will try to restore fully, but some of them may do it partially, especially I think if I am right, this non-homologous end joining, it may do like that a little bit. So this also you keep in mind, we will go one by one. It's very simple, it's not that difficult, but I already asked this question, is there any advantage having many pathways? Yes, when there is a small problem with one pathway, another pathway can partially compensate. Sometimes if the one pathway is, what this one talks about is partial defect and complete defect. So if it is completely absent, now starts depending on the other pathway. If it is only partially defective, it will partly depend on that pathway and another part it will use the other pathway. If it is fully defective, it will fully depend on the other pathway. It's simple. Just use your common sense, it should come. DNA repair pathways are obviously highly regulated. The more number of these things are there, more complications come. So naturally there will be more number of problems. We say that when we have 1 billion people, we have 1 billion problems also. So it is like that. You will have more number of pathways means more number of problems also to tackle. So they are highly regulated. DNA repair pathways are differentially active in various tissues and cell types. I hope you understand this. Just like differential gene expression, these may not be the same in every tissue. Kidney tissue may have certain pathways, liver tissue may have certain pathways, some other tissue may have some other pathways. Depending on that, its vulnerability to become cancer will also be different. Because DNA repair pathways are very important for the cell to become a cancerous cell, for a normal cell to become a cancerous cell. How efficient, like our immune system. If our immune system is very good, then we are unlikely to get certain diseases, right? If we are weak, partially weak, you will get to some extent. Fully weak, you have more chances of getting. Similarly, this also DNA repair. So it differs from one tissue to another. Now we will talk about the so-called base excision repair. This pathway takes care of damaged DNA bases or single strand DNA breaks usually. The pathway is useful when there is a spontaneous DNA damage, such as what I told you, DNA deamination, hydroxylation of bases, or the damage by alkylating agents. DNA glycosylases, these enzymes remove the bases that are damaged. This results in the formation of what is called because bases are either purines or pyrimidines. If you remove the purine base, then such a condition is known as apurinic. A means absence. Usually the terminology is apurinic. If there is no pyrimidine, that particular site is called AP site, not your Andhra Pradesh. Apurinic or pyrimidinic site. So AP site. APE APE1 endonuclease specifically recognizes sugars without attachment. See, once the base is removed, then that is recognized by certain enzymes, because normally the base is attached with the sugar and then phosphate. Now in this case the base is removed, so sugar stands alone without the base. So in such cases this endonuclease comes and recognizes and it acts on, it forms the so called 5' deoxyribose phosphate. DNA polymerase fills the, I think if we read these sentences you may not understand it fully. We will look at the figure, it will be easier. See this. Let us say there is a DNA damage in one of the strands. Now what happens? There is a cut here. The base is still sticking, but it is chemically modified, the base, right. Now the damaged base is removed by DNA glycosylase. That is what I told. It recognizes that this base is not linked with sugar, so it removes it. It removes the base. Now the next one, deoxyribosyl phosphate exercised by the APE enzyme. So it creates a gap completely, because otherwise you see the attachment, right. So it now makes a real break. There was only a damage, now it really breaks that chain. Why it breaks the chain? So that it can be filled. So that you need polymerase and then ligase. With that sequence it can be filled, the gap can be filled. Now you see between this the first one and the last one almost, I mean like except for this modification, if you imagine the original one, you are restoring the original structure. So you are ultimately restoring the original structure. So this is what is called base excision repair. There is another kind of problem which is called mismatch repair. As I told you again, if you look at it here it will be better, because if I start reading those sentences you may not get the clear picture. So you have some error, so that there is a base mismatch. Base mismatch you understand, A base pair to T and G base pairs with C. If there is a mismatch there, for A there is no T here and there is something else, maybe C is there or whatever. So there is a mismatch. So in the newly made strand, if there is a mismatch, then immediately there are what are called proofreading proteins. They come and attach. They are called MUT-S, MUT-L. These were discovered in bacteria and other organisms. So they come and attach here. They come and bind. See, basically in biology there is another thing that you have to understand. Whenever there is a structural change, some proteins will come, and in the immune system also I told you, P53 if it is getting mutated, then it will be seen as a foreign material. That is how the immune system recognizes it, and that is how we are protected. So whenever there is a change, something will come and bind, not necessarily the immune system alone. Some proteins will come and bind. Today the AC is less. Usually it will be very cold. Anyway, I don't see anybody there. Okay, anyway, we will see. Even I am getting the throat choking. In either way, if there is no AC or if there is full AC also. So okay. Now what happens, like the proofreading system, the scanning of the DNA detects the nick in the new strand. So ultimately what happens, certain enzymes, they will cut that portion. something like 15 to 25 nucleotides or so will be cut off. Wherever some defect has happened, wherever there is a mismatch, in one of the strands, it will be completely cut off, so that you make a break, keeping the other strand intact, because you can later on, like I told you, the complementary strand is there. So, now you can synthesize with polymerases and ligases. So, that is what is happening here. Now, we will go back and look at some of the names. So, DNA replication, as we know, it is prone to errors, and these errors in the form of mismatched nucleotides are mainly corrected by this kind of pathway. It also detects and corrects DNA adducts, such as those formed upon treatment with platinum based chemotherapeutic agents. I just told you, cisplatin, oxaliplatin, all those kind of things. So, many genes I mentioned already, the abbreviation is MUTES homolog, MUTEL homolog. That is why you have these names MLH1 and PMS2, MSH3 and so on. There are number of proteins. They are all involved in mismatch repair. What is their role? They recognize this kind of a mismatch and go and bind in that place. So, that is their role. The heterodimeric, they will form heterodimeric complex, they will recognize the nucleotide mismatch, and this gives rise to some interaction with other complexes. Nucleotide excision and resynthesis obviously involves many enzymes and proteins. If cancer cells are deficient in mismatch repair, they will have, I kind of asked you that question. If the cancer cells have defect in DNA repair, that is good for them. I mean, not really good, like they will have more kind of more mutation frequencies than normal cells, because mutation will be prevented by this DNA repair mechanism, and if you do not have this mutation repair mechanism, then it is easy to get more and more mutations. So, in that way, as I told you, it is advantageous. You have to think from the point of view of cancer cells or normal cells at a given time. Once you have that thinking, then you will not have confusion. Otherwise, we are bound to get confused. Even I get confused sometimes, because the student sometimes misleads me into asking something, and I also forget for a moment, and then the answer is wrong. That is how I have noticed over the years, the confusion is mainly because one of the persons, it can be a student or it can be teacher, who misunderstands the situation or does not understand from what situation he is talking about, whether he is talking for the cancer cells or is he talking from the point of view of normal cells. If you have that kind of a thinking process, then you will not get confused. So, here we are talking about cancer cells. If they are deficient in mismatch repair, they will have a much higher frequency than normal cells. So, that is the idea. Such cancer cells exhibit what is called, you should know what is microsatellite instability. So, this I have kind of explained. Then what is called, what is this full form? Nucleotide excision repair pathway. This pathway is, that is, each pathway has some role, as you can see that this one was for this natural errors and for DNA adducts. The other one was for single strand DNA breaks and for alkylating agents. This is very important. Which pathway is suitable for what, that you have to kind of remember. But mostly I can tell you, so far whatever we have discussed is usually for single strand break. Only when we go to that homologous methods, non-homologous and enjoining and all that, that is more suitable for double strand breaks. So, the first three are single strand breaks, next three are probably for double strand breaks, like that you can remember, just for remembering I am saying. Okay. Nucleotide excision repair, this pathway takes care of DNA adducts. The problem is there are so many proteins, it is difficult to remember. Like I told you, MSH and all that, they are involved in the other pathway, but in this one it is called XPA, This is not done by one protein or something. Hundreds of proteins are involved in these repair mechanisms. So it is called XPA, XPB. These names again are a little bit headache because they have been derived from some other lower organisms, and those names we may not be familiar with, and so on. Anyway, this is another one, X, P, A, B, C, D, E and G. These proteins are involved. XPF, what is called ERCC1, are also required for the processing of DNA cross-link repair. These proteins, they will cooperate to recognize and excise cutting the damage. Usually the idea is, if something is damaged, first just like we are doing the wound cleaning and all that, when you have a wound, that portion, the people will first scrap it. Actually they will cut and remove and bring it to one shape and then give some medicine, something like that. You clean up the area. Similarly here also, wherever that portion is there, they will cut off that DNA base pair and all that, so that it is completely cut off and you create a nick. Now it will be easy for you to repair because you have the other strand. So as far as single strand breaks are concerned, if there is a partial nick in one strand, you better cut some 10 bases, 20 bases, so that you make a gap. Now you will have space for synthesizing them, DNA synthesis. So that is how it is done. If you understand the overall logic, it is easier for you to. For that you need DNA polymerase, regular method how DNA is synthesized you should know. DNA polymerase, DNA ligase, all those things, they are required. Similarly, a DNA, another thing that you may have to remember, whenever there is a nick, whenever there is a structural alteration, some proteins will come and bind. So it is our job to remember what proteins come and bind and recognize. So there must be some proteins to recognize, there must be some proteins to cut, there must be some proteins to heal. That is all is the DNA repair mechanism as far as the single strand breaks are concerned. Only difficulty is in remembering the names of those proteins that are recognizing this. In each pathway it is different. That is the only problem. So here DNA binding component was so called DDB initially binds to the site of damaged DNA, such as here in this case cyclopyrimidine dimers or 6,4, yeah, we have talked about this 6,4 photo product and all that. I already told you how the UV radiation can create thymine dimers and all that. So the so called DDB is a part of ubiquitin E3 ligase that polyubiquitinates so called XPC. This is very important molecule XPC. Polyubiquitination of XPC results in enhanced DNA binding. Here we are talking a little bit deeper about how the DNA binding occurs, how the site is recognized, how the DNA binding occurs. We will look at it. There is another complication in this pathway. What eukaryotic pathway especially has two components, what is called transcription coupled repair and what is called global genome repair. Global genome means you are talking about the whole genome, so it is naturally a slow process, random process of inspecting the entire genome, because I talked about inspecting the genome, scanning that and all that. How far you want to scan? Do you want to scan a small portion or you want to scan the whole genome? So depending on that you divide it into two. That GGR, global genome repair, is a slow and random process because it inspects the entire genome for injuries, for damages. Whereas the so called transcription coupled repair is specific, efficient and concentrates on damage blocking RNA polymerase II. So where the RNA polymerase is there and there is some damage around that area, only in that area it will concentrate. So these two mechanisms differ in substrate specificity. If we go deeper, it is still more what are the substrates and all that, I don't remember also, and that is much more detailed, it is not necessary actually. These two mechanisms differ in substrate specificity and recognition, and hence the enzymes involved are important nodal points for many post-translational modifications. I realize that it may not be that easy, but what I suggest is that at least remember the names of the pathways and remember what for they are useful, under what conditions this pathway operates, and what are the proteins that are involved in that. Further mechanisms are not necessary. Again, a picture tells a better story. So, we will look at it. See here, some adduct is there, helix distorting. I told you when there is a such adducts, there will be a kink and other changes in the helix. So, first that is formed, then that has to be recognized. Once it is recognized by those proteins that I mentioned, then you cut that portion, as I told you, approximately 24 nucleotides from 5' side, 5' side of the adduct. Wherever the adduct is formed, from the 5', about 24 nucleotides are removed. Now resynthesize the DNA in the normal 5' to 3' direction, and finally ligate it with a ligase. So, same manner it is done. So, this is what usually done by nucleotide excision repair. I think we will kind of stop here because there are 6 pathways. Now, I told you those 3 are single strand. Now, we have to talk about double strand breaks and how that is repaired. We will do it in the next class.